So when we assemble an RNP complex, it's actually very simple. We have two tubes. You have one tube that contains your Cas protein, and then you have a second tube that contains your guide RNA, which will then guide your Cas protein to your specific target in the DNA. What you do is you take a specific volume of both of them, you put them together, and actually leave them on your bench for 20 minutes, and automatically the RNA will be incorporated in the protein and your RNP complex is formed. The RNP complex delivery in plants, you have a few options. So you can, for example, bombard them literally into your plant cells. So then you coat your RNP complex usually on gold particles, and you blow them at your tissue with high speeds. What I'm more used to doing is actually taking a chemical called PEG, or polyethylene glycol, and which softens the membranes of the plant cells and actually carries the DNA inside the cells. So those are two possibilities, but there are others. For example, with an electroshock, you can electroporate some cells and allow the complex to enter. So we have different possibilities. Sometimes it depends on your specific species, what works and what doesn't work. We can use agrobacterium to deliver DNA into cells, but not RNP complexes. So RNP complexes are really the isolated protein of Cas9 with the guide RNA. But of course, we can make a vector that contains all these elements, so genetic elements that express Cas9 and genetic elements that express the guide RNA. And then we use agrobacterium to bring this DNA into our plant species of interest. This DNA will be incorporated in the genome of that plant, and then the cellular machinery will allow this to get expressed, and then you have your in vivo assembly of the RNP complex. The main reason why I would choose for agrobacterium is that you have a higher chance that you only have one copy of this foreign DNA in your genome, compared to, for example, bringing in DNA via biolistics or electroporation. I would prefer RNP delivery, so the purified protein, if I would be very concerned about off-target effects, for example, because this protein only lives for a couple of days in your cells. So basically you bring it in, it does the edit, hopefully, and then it's just degraded by the cell itself, and nothing remains in there. Whereas if you bring in DNA, of course your plant is mutated and becomes a GMO, and you cannot get rid of that DNA so easily. You need some crossing and some advanced generations to do that. Technically, assembling an RNP complex is very straightforward, but one of the limitations might be cost. Buying a purified Cas protein is pretty expensive. Also, you can buy your guide RNA ready-made. So these things cost a bit, and depending on how large your experiment is, how many genes you want to target, this can blow up very easily. So in that case, if the costs become too much of an issue, maybe a DNA-based delivery of your vectors might be a more cheaper solution. For delivery of the RNP complexes, we do not really run into much problems, I have to say. But of course, some plant species can be transformed very easily. Some plant species can be transformed a bit more difficultly or need a very specific protocol. So it really depends on your model organism, if you can have some issues with transformation or not. Some plant species get really low transformation efficiency, so only a couple of cells in your Eppendorf or in your population will be transformed. If you have a very complex genome, the delivery can stay the same, but you might run into problem finding a perfect target. If you want to target only one specific piece of the genome, some plant species have 3 different subgenomes which are very alike. So then if you only want to target one specific subgenome, it can become tricky to find the perfect guide. So most plant species, the transfection efficiency, transformation efficiency is not too bad. But for example, I know in my seaweed in Ulva, I usually only get 1 out of, let's say, 1,000 cells that gets transfected, that gets these molecules in, so which is at the low end of things. So if your transformation efficiency is very low, and this happens in some plant species or in my seaweed, then you actually need a very good selection system. And we usually screen for resistance to certain types of chemicals, for example, or antibiotics. So only plants or seaweed that contains a specific edit will be able to grow on a very specific growth medium containing a specific antibiotic. So when we have delivered the RNP complex to plants, basically what we want is to enrich for the plants that have a mutation. So there are certain ways to do that, but one of the most straightforward ways is to screen on a specific chemical. And so, for example, only plants containing a mutation will be able to grow on a specific herbicide, or will have a very strong visual character. For example, the lack of trichomes if we mutate this glabra gene, or containing white spots or white regions if we mutate a PDS gene. So this, especially if you're developing a technique, these types of screening methods are vital. And then, of course, if you have something that grows on your medium, you want to confirm that the DNA is really edited. So what we do is we take a sample, or we take the entire seedling if we don't need it afterwards. We purify the DNA, so the genomic DNA of this plant, and we will amplify a specific part of the genome, and of course the part of the genome where we hope to have made a specific mutation. When we amplify this using PCR, we actually purify this PCR product and send it for Sanger sequencing. So Sanger sequencing will read the entire code of this PCR product, and then using some bioinformatic tools, we can see if we have a mutation, which percentage of our cells are mutated, so the type of edits we have made. All of this is then the bioinformatics work that comes afterwards. So the efficiency of CRISPR, again, depends a bit on your specific system and on your guide RNA. But let's say in our model organism with a top performing guide RNA, you can have easily 80% of your primary transformants which are mutated. Then depending on maybe choosing a lesser active Cas protein, it can become 0. So it is really sometimes trial and error. What is often also very crucial is that the mutations that you induce are also transmitted to the next generation. I'll explain this. In Arabidopsis, for example, if a seed germinates, some cells might be mutated and some might be normal control wild type. So when the plant makes a flower, you actually want that the cells who make the flower and make the seeds for the next generations actually carry your mutations and do not contain the control or not mutated DNA. So sometimes we do not, usually actually, we do not only screen in the first generation, but also in the second generation if the plants contain the mutations.